So next up, we have Dr. Abhishek Banerjee from Newcastle University. Abhishek, please take it away.
Okay, I'm unmuted. Can you hear me all? Great. All right. So thank you very much for the introduction. I would just like to quickly sort of move on. It's nice that Rafiq presented his talk right before me, because he mostly talked about sensory motor decision making that are based on sensory motor transformation. So here I'm going to talk about a slightly different thing, which involves sort of certain cognitive switches. And of course, the theme of the talk would be quite similar, which is the theme of the symposium, which is the prefrontal control and gating of flexible sort of sensory processing. And I have also started my own lab recently in the Newcastle University in the UK. So when we talk about sort of value-guided decision making, we generally talk about inferring meaning of not the sensory stimulus itself only, but also what this sensory stimulus outcome is. So perceptual decision making, as we know, or goal-directed learning, it involves multiple cognitive maps, and these maps are both cortico-cortical, cortico-subcortical maps. They are constantly talking to each other in a distributed brain region. But animals do not only make decisions. They evaluate and switch between decisions, between appropriate sort of goal-directed strategies, and they do so by a method, sort of a way of depicting this would be sort of a stimulus action outcome evaluation. So it's sort of less clear how prefrontal neuronal populations or subpopulations actually encode decision variables during a flexible decision-making task, and how their responses flexibly remaps from one sensory outcome modalities to another ones during value-guided decisions. And also another very interesting question is, we know some of these prefrontal regions, including SEC that Rafiq talked about, other areas such as orbitofrontal cortex, medial prefrontal cortex, they sort of encode decision variables, but how they specifically control primary sensory areas in a sort of a hierarchical, guided, reinforcement-driven manner to support value-guided behavior is sort of unclear. So what we did in order to address this question is we designed a task that's based on sort of reversal learning of a stimulus outcome association. So reversal learning involves adaptation of behavior according to changes in stimulus outcome contingencies. So what you see here on the left is as a mouse, again, a kind of a 2 months old, 2 to 2.5 months old mice that are, again, head fixed. And we are presenting them with distinct tactile cues. So different types of sandpaper textures that you see here. Like this is a coarse sandpaper. This is kind of a fine sandpaper that they have to distinguish in order to, they are water scheduled animals in order to win water reward. They're head fixed, and we are measuring other behavioral variables. But as you can see, since it's a go-no-go task of a tactile discrimination-based go-no-go task, the trial structure is somewhat like this. You have a baseline time when the animal has to sort of wait, and then there is a stimulus presentation. In this case, a tactile sort of cue comes towards the animal. The animal is to make a choice between a go and a no-go stimuli, and lick or withheld licking in order to implement, in order to win a reward or to avoid a punishment. So as you can see, during a go-no-go task, you have 4 different outcomes. If you have a target texture that leads to a winning of a reward, which is a hit, and they can miss the target texture, which is a miss, followed by a false alarm when they incorrectly make a decision, and correct rejection when they correctly avoid the non-target texture. So if the animals, sort of we train the animals individually on this task, and this is a single mouse that I'm showing here, if you can see that the animals actually learn the task quite nicely. Their hit and correct rejection rate over the days of training actually increases and almost reaches the 100% level. And their false alarm rate or their errors actually go down during learning. So in order to understand or study flexible decision making, what we do now is once the animal learns the task perfectly well, we switch the rule, which means if the texture one was giving reward initially, they learned this outcome stimulus outcome contingency. After they have learned that particular stimulus does not give reward anymore, it becomes a no-go. But texture two, a no-go texture, now gives them reward. So, and as you can see, a single mouse actually relearned the task fairly well. If you see a bunch of animals, they can also relearn the task from learning through reversal. And each of these phases undergoes two different phases themselves. One is learning naive through expert and reversal naive through expert. So animals actually learn this sensory discrimination task, a cue guided task, and they adaptively change their behavior. So this is a pure sensory task. And what we were interested in is to really look at some of these prefrontal areas that play an important role in cognitive processing of information or stimulus outcome associations, invoking rule-based strategies, if they play an important role in these type of tasks. And I can cut short and right away tell you that selective silencing of a very particular brain structure in the prefrontal area, which is the lateral part of orbitofrontal cortex, impairs reversal learning. It has been seen in other modalities as well, in other animals as well. It's a very particular structure in the brain that plays an important role in stimulus outcome association. So OFC is a key higher order association cortex that has both projections to subcortical structures as well as sensory cortices to implement these type of learned association related rules. So what we did is, so as you can see from Marie Carlin's review, the OFC or the orbital areas are located quite deep down in the frontal part of the cortex. So in order to understand what is happening in the brain, we created or we prepared a novel preparation through which we started imaging these neurons using two-photon imaging through a green lens that was sort of inserted through a metallic cannula and a cover glass that's at the base. So we are imaging lateral orbital cortical neurons throughout behavior. So how do these neurons respond? So this is an example neuron. You can see that I have aligned their trial-related calcium responses according to the learning and reversal. So I asked you sort of to remember that the learning naive through learning expert is the behavioral sort of output, and reversal also has a similar reversal naive through reversal expert. So I guess what you can appreciate from here is one very interesting thing happens as soon as we switch the rule, which is right this white line that's here. So that most of these OFC neurons that you can see the population average responses here, a lot of these OFC neurons, including the example ones here, starts responding to the newly rewarded context. So if whenever there is a mismatch of a stimulus outcome evaluation, through evaluation, stimulus outcome contingency, these neurons starts encoding that sort of a value mismatch or value prediction error responses. It's not seen for the correct rejection responses. It's very specific to the hit responses that are leading to a reward. Now, since we are doing two-photon imaging of individual of these neurons in OFC, as well as in the primary sensory cortex, of course, that you can see the sensory neurons in comparison with this predictive responses in reversal naive phase for OFC, it shows actually an opposite pattern of responses. Sensory neurons in a very similar way, it's as primary sensory neurons, shows a learning induced responses. But then as soon as there is a rule switch, there is not much happening in the sensory, primary somatosensory cortical neurons. But they completely remap their responses at a later stage during training when the D prime or the behavioral scoring has gone up again. The animals have relearned or switched the behavior. So this is extremely interesting that on a same task dependent manner, there are two areas that are completely talking in a very different way. Now, since we record these neurons longitudinally through a cortical window preparation and a green lens preparation, you can really study all of these individual neurons, hundreds of these neurons individually. And I want to highlight two important things here. One is the response alteration. So you can really study how during distinct behavioral phases, all these different neurons are being engaged or disengaged, right? So I want to highlight two things here. These two cell classes of the populations. These are outcome selective neurons and gain selectivity neurons. So how do we measure this? So what we did here is we plotted the responses of these neurons before and after of a reversal. So as you can see, the most important bit for OFC neuron was this transition between learning expert to reversal naive, when cells are mostly responsive. Whereas for S1, the transition between learning expert to reversal expert is the most important phase, because the remapping has completely happened at that phase. So here what we see is these outcome selective neurons that are responding in both cases before and after reversal. So they are the ones that are there understanding this value mismatch representation, and they can actually encode or guide the rest of the brain perhaps to understand that there has been a different stimulus leading to a different outcome, and they are understanding this encoding. So if we now look at plot these comparison between learning expert reversal naive through learning expert and reversal expert, if we plot these neurons individually, and I would like to highlight the blue and the red neurons that you see here. When we look at the somatosensory cortical neurons, these are S1 neurons, primary sensory neurons. When they are if we look at their responses, what is extremely striking is during this transition from learning expert to reversal naive, immediately after reversal, when the S1 neurons are not so much, they're not so sure about how to respond, their responses are—
Abhishek, you have 1 minute left.
Yeah, great. So they're all sort of around these 0 points in an ROC plot. But all of these same neurons actually completely remap when the animals relearn the task and goes to the next level when they become reversal expert. Now, our guiding hypothesis is there is this massive remapping of reconfiguring of responses in primary somatosensory cortical areas. So our hypothesis was, if we silence orbitofrontal cortex, silence or remove these value prediction error responses from the lateral OFC, whether a similar kind of responses arise in the primary somatosensory cortex. So we silenced OFC, a lateral part of OFC, using DREAD receptors, and we silenced OFC after reversal or during rule switch, right after reversal. And I guess what you can appreciate here is to show you this marginal distributions of these cells. So this is the reversal learning expert to reversal expert marginal distribution. I'm plotting it in a slightly different way here. So this is S1 neuron. The pink one is reversal naive through, red one is reversal expert. There is a massive rightward shift from the previous contingency -1 to new contingency +1. This is in primary somatosensory cortical neurons without OFC silencing. If we do now the same experiment and silence OFC and record from the same S1 neurons, you see there is actually a complete overlap of these two. There is no rightward shift, no shift towards the primary somatosensory cortical neurons towards the new contingency. So this basically sums up really what I had to say, which is really during sensory processing, the sensory input, sensory learning dependent responses in primary somatosensory cortex builds up responses through this feed-forward mechanism in the lateral OFC, which is a key area for value-guided decision-making. And it computes, subpopulation of neuron computes this value prediction error responses. And through this teaching signal, feedback teaching signal that's coming from OFC to S1, together with many other areas, I should point out, that actually implement some of this responses. Just to sum up again, OFC is an important—
Abhishek, we are running out of time quickly, so can you just—
This is the last slide, so
okay.
I'm going to end. So OFC is a key hub for value-guided decisions. Silencing OFC impairs reversal learning, and neurons in the OFC actually plays a very important role providing value prediction error responses. And a functional subpopulation of these neurons that are outcome selective neurons that I showed in OFC and S1 signal reward expectancies by integrating the reward history. So I'll stop here. You can see more of this on this recent paper that we published. And I would have to acknowledge Fridjof, whose lab this work was done in Zurich, and my students here in Newcastle and also in Zurich. Thank you.
Thanks, Abhishek. That was a very exciting talk. So we have some questions.
Mhm.
Oops. The first one is from Lawrence. Do your neurons in OFC have mixed selectivity for the different features of the task, or are there distinct clusters of neurons which are selective for individual features?
That's a very good question. We do have very distinct types of neurons. There are some neurons that actually encode for sensory features as well. But a major fraction of these neurons are actually outcome selective neurons, which is kind of sort of expected. But we are showing it for the almost, I guess, for the first time that individual neurons are encoding these type of features.
Perfect. So the next one is from Mehmet. Great talk. What happens if you inactivate ventral OFC in this task?
We have silenced the medial part of OFC. We have, and it does not have any effect on this particular form of reversal task, but it plays an important role in other forms of reward-driven learning.
All right, great. Thanks again for the wonderful talk. Applause on behalf of the attendees. And next up, we have Philip Cohen